Allow me also take this opportunity to welcome you back to this other session. Now we are going to look at the relationship between photosynthesis and the synthesis of food in green plant. That's going to be our main area of emphasis today. Now, let me begin like this. We are saying we are beginning from where there is rubib. We say that rubib is a carbon dioxide acceptor molecule. In presence of carbon dioxide, it forms what we call the glycerate 3-phosphate, which is the GP, and this occurs in photosynthesis, or what we are calling the Kelvin cycle. Then the GP, or the glycerate 3-phosphate, divides into two, the way you can see here. One is the glyceraldehyde phosphate pigalo stroke the GP, that is the glyceraldehyde phosphate, and the second one is the pyruvate. Now when you look at the pigal, that one divides. So this one forms what we call the glycel. Then the glycel, from the glycel, it forms what we call the lipids. Then the pyruvate, these ones break into what we are calling the fatty acids, which forms the lipids, and the pyruvate still combine with the coenzyme A to form the acetyl enzyme, the acetyl coenzyme A, which enters into the Krebs cycle. Then from the Krebs cycle, this one forms what we call the alpha ketoglutarate. This part is the so-called alpha ketoglutarate. Then here we are saying that inorganic ions uptake from soil, they form the nitrate, then ammonia. So when ammonia combine with the alpha glutarate, we saw that we shall form what we call the glutamate, this one. Then this glutamate goes under that process of transamination. So we looked at this when we are looking at photosynthesis to form other amino acids. So this one is the one you are seeing here. Then from there it goes down and forms what we call the proteins. Then another route. Now we are seeing that now let us look at the other route. Now the back to the what? To this route of the glyceraldehyde phosphate, or the pigal stroke, or the TP. Now this one goes back to form the rubib. Then from there the other one goes and forms the 6 carbon, which is called the phosphorylated sugars. Here you have one is monosaccharides, where I have glucose, fructose, disaccharide, where we have the sucrose, and the polysaccharide, where we have the cellulose and the starch. I looked at that. Now here is some question where they are saying that the enzyme Rubisco, in spite of being the most common enzyme in the world, so when they can ask which is the most abundant protein, is that Rubisco. Now is very insufficient in photosynthesis. Explain this statement. One, we are saying that the Rubisco can add approximately 3 carbon dioxide to 3 molecules of rubib each second, which is very slow for an enzyme to make up for this. So plants produce large quantities of Rubisco, with it composing 50% of the protein in a chloroplast. Now too, we are seeing that Rubisco is not a very specific enzyme, as it sometimes combines the rubib with the oxygen rather than. We are seeing that this Rubisco can sometimes combine, rubib can combine with the oxygen rather than what? Carbon dioxide, because of a relatively non-specific active site causing photorespiration, which leads to formation of useless oxygenated intermediate rather than carbon dioxide fixation. Now let us look at what we are calling we are saying photorespiration. We are saying that oxygen of rubib by rubib oxygenase, in bracket rubib carboxylase, at high temperature, low carbon dioxide and high oxygen concentration to form the phosphoglycolate, which undergoes oxidation in the peroxisomes and metabolism in mitochondria to release carbon dioxide, thereby preventing carbon fixation in C3 plants. Then we are saying that how photorespiration affects plant. We are saying that when C3 plants are exposed to low carbon dioxide concentration or high oxygen concentration, e.g., when the stomach closes to reduce water loss, rubib carboxylase catalyzes the reaction between rubib and oxygen to form a 2-carbon compound, so which is the phosphoglycolate, which is oxidized to release carbon dioxide. Yet when the carbon dioxide concentration is high, rubisco enzyme catalyzes the reaction between rubib and carbon dioxide to form a 3-carbon compound, which is the 3-phosphoglyceric acid, PGA, which undergoes several reactions to form sugars useful to the plant. It is estimated that photorespiration therefore reduces the potential yield of photosynthesis by 30 to 40%. I therefore sign out. So I'm just waiting for your question in the comment section. So time is now or never. I sign out. Yeah, we shall meet in the third part, which is the hack, the heart tracker cycle, or what we call the C4 pathway that has been C3. Yeah, I sign out.